So welcome back guys. So after New Year we are starting our next video lectures. So today we are going to start about the CRISPR Cas9 system. Now this CRISPR Cas9 system I have searched internet and also I have searched some study materials from it about this actually. So this process looks very complicated, but it is not. Okay, let me mind you that it is not that complicated. Simply think of bacteria being normal humans. Like in case of humans, we have got our immune system. The bacteria also has got some kind of immune system within it to protect it against different kind of viruses. Actually, this is specifically designed for the phages, which attack bacteria and harm them, and it is not good for them. So it's specifically designed for this kind of phages only. Now, this CRISPR system consists of CRISPR-Cas9 conjugate. Now, this CRISPR-Cas9 conjugate or this whole system is encoded by an mRNA. Okay, and it is found in the genetic loci, that is CRISPR loci. From CRISPR loci, we are getting all the components of CRISPR. Now, procedure-wise, this is actually a RNA mediated, actually I can say RNA guided adaptive immune system. Okay, it is encoded by the CRISPR loci, just I have said now, and it accompanies the CRISPR associated genes which are named as this Cas genes. Okay, now how it helps? It basically provides bacteria with some immunity. Immunity against what? Immunity against bacteriophage infection, or sometimes it is also immune for plasmid transfers. Okay, now starting with the CRISPR, we have to get this in our head properly. That is, CRISPR contains the thing CRISPR. Okay, the full form I will not tell. Make a Google search, it is very easy to find it out. And we have got a Cas9. These are all conjugate complexes. Okay, so this CRISPR is actually a short generated 20 nucleotide long to 30 nucleotide long recognizable viral DNA. Now this viral DNA are usually recognized by the help of something known as PAM sequences. I am going to talk about the PAM system a bit later. Let's first start it with this system. Now over here I have drawn a genome, basic DNA. Okay, now I am saying that this is a foreign genome. So I am meaning this foreign genome as this can be a viral genome, this can be a plasmid, this can be anything. Okay, sometimes even this type of things happen when we are introducing some new genomes in the system. Okay, so this foreign genome, as I have said, this foreign genome is actually broken down, actually not broken down first of all, but there are some PAM sequences. This is where I am pointing my laser, is actually a PAM system or is a PAM site. This PAM site contains specific sequence which is known as the PAM sequence. What it does is that this PAM system, as I have told, is only present in case of viruses. Okay, so and this entire whole protein complex is actually the CRISPR-Cas9 complex. Okay, or as I call it, the Cas9. Now I am not calling it Cas9 because I can call it so like that, but it's a general it's a bookish term obviously. It acts as a spy. What does a spy do? It sends messages to the people. Okay, it spies about the people. Exact function of spy I cannot elaborate like that, but it actually helps in passing or communicating about some things like political things or anything. Okay, so now this CRISPR-Cas9 system contains a 20 nucleotide sequence which is actually complementary to this viral genome. Now this complementarity arises because suppose your body has got some infection, first-hand infection. So as you are getting cured because of your immune cell or some kind of drug thing, I don't know, you are actually leaving some memory cells. Okay, you are developing some memory cells which I am going to cover in some later videos. Obviously those memory cells are retained in your body and those memory cells are helping you to fight it off when you are again developing that infection. So maybe some prior infections have resulted this formation of this 20 nucleotide sequence, which is basically a RNA sequence, the complementary RNA DNA hybrid sequence. This is the DNA, foreign genome DNA, and this is an RNA. This red thing is RNA over here. So these RNA complementarizes with the DNA and helps in recognition of sequences which are just beside the PAM sequences. As it does so, as we are knowing this as CRISPR Cas9 system, Cas9 is a nuclease. Okay, what kind of nuclease? It basically degrades DNA portions. So it's specific for DNA only. So this Cas9 degrades the DNA from the point where it recognizes this site, 20 nucleotide complementarity site. Now, if this is a foreign genome over here, this foreign genome is split open by this CRISPR Cas9 as soon as it identifies the PAM. So there is a particular subunit on the Cas9 CRISPR system which identifies this PAM, resulting in the formation of this kind of structure where it basically cuts off the portion of the viral genome. Now this cut off portion can be utilized by it for different many purposes in the bacterial cell, depending on what kind of condition it is facing outside, and also they may have different functions, okay, which I am not going to discuss in details because it can increase the complexity of this video. Now after this cleavage from these sites, it's a double strand breakage, double strand breaking double strand break thing forms. I'm not getting that proper term right now, but basically it induces double-stranded breaks. Okay, so these double-stranded breaks, what it does is that these double-stranded breaks are formed in the DNA. So particular parts of the genome is being taken out or being cut off from the part. Okay, now this will help in two things. This will help in two ways. The viral genome is now rendered useless, and hence it can be it is not that much functional in the bacterial cell. In the other prospect, how we can use it. We humans always try to take account of natural systems and modify it in such a way that we can actually make a different system utilizable by us. So what we do is that we actually induce this kind of double-stranded breaks. Now this Cas9 identifying this particular sequence of the PAM sequences is basically done by something known as the sgRNA. Okay, this sgRNA is nothing but it's basically a guide RNA. Okay, or as I like to say in case of RDT, synthetic guide RNA, sgRNA. So this sgRNA, we can make this sgRNA so that it can particularly go and bind to some kind of sequence in the genome, and thus the Cas9 present in the whole system can cut that genome out. So let me help you understand this first. What happens is that first of all we have got the PAM sequence. Whenever it encounters the PAM sequence, this DNA is entitled to be destroyed. First point, remember. Second point, as it was the PAM sequences using the 20 nucleotide sequence over here, what it does is that it tries to break this, okay, inducing double-stranded breaks like I have shown over here. Now these double-stranded breaks, as I have shown you, is caused because of this Cas9 protein or spike Cas9 protein, as I have told, because of nuclease. Okay, now as I am inducing these double-stranded breaks, things can happen. Now these double-stranded breaks are not if you are making a synthetic sgRNA, then your sgRNA will actually bind to specific part of the genome which you want to silence or which you want to edit, and that can be done by using the sgRNA over here. So this sgRNA, what it does is that it particularly helps you to edit or delete that part of genome or silence that part of the genome and insert your new target DNA over it. It can happen, or otherwise if you are just planning for silencing, that happens that double-strand breaks are induced, which can be repaired using non-homologous end joining, causing sometimes it causes random mutations. It is prone to mutations basically, but we have also got homology dependent direct repair or homology direct repair or dependent direct repair, which is also known as HDR. So this HDR is nothing but homology dependent repair systems, which I am going to cover in some later videos. But obviously this is the CRISPR Cas9 system, so I am summing it up for you guys. It is very simple. So what happens is that we have got some PAM motifs in the viral genome. Okay, one question may come, that is this PAM sequence, what is this PAM sequence actually? So this PAM sequence, I have not written it, okay, I forgot to write everything. But this PAM sequence is basically a 5 prime, from the 5 prime, and if I'm starting, it's a 5 prime nucleotide, it can be anything, followed by 2 side to back guanine residues, okay. And it's basically I'm talking about the usual PAM sequences. But if you are talking about some non-canonical PAMs, okay, if you are trying to design some non-canonical PAMs, then the sequence is quite different. Okay, my camera angle is tilting, I guess, because I have taken it and kept it at the side of a book so that I can support, but it's getting tilting. Okay, so also we have got some other kind of canonical PAMs, or which is 5 prime, a nucleotide followed by a guanine and an adenine residue. So these are all PAM sites. Okay, and actually we are also working on this Cas9 systems, and not we, actually many scientists. I am not a scientist, I am a layman, okay, and I do this CRISPR things, okay. So this Cas9, what it does is that if you are trying to edit this Cas9 in such a way that it can easily edit any genome, it can easily recognize any PAM sequence. If it recognizes any PAM sequence, then we can edit any kind of genome, okay. So it's a very huge kind of research going on nowadays. So application part I am going to deal right now. So application is first of all gene targeting. Gene targeting means if you are targeting some specific gene using the sgRNA, so you have to know the gene, okay, this is a thing. You have to get a brief idea about the gene, and then you have to design the sgRNA such that it binds to the gene of interest. Drug screenings, okay, so many drug screenings are also done using this kind of CRISPR-Cas9 systems, but useful is that genetic screening. Many genetic diseases can be screened by using the CRISPR-Cas9 systems. And also we have got the ligation mediated genome editing. So ligation means ligase, like ligase type of functions, okay. So ligase mediated genome functions can also be done using this kind of CRISPR-Cas9 system. Now this is just a 8.4 kilobase pair part I am talking about. Is basically a kind of plasmid I am trying to draw over here. It's nothing but it's just a plasmid containing Cas9, some CMV along with the sgRNA, okay. I am not going to details of the components except for Cas9 and the sgRNA and the target DNA. These are all part of it, and we already know the ORI and the AMPAR condition. AMPAR means amplicity resistance and ORI means origin of replication. So we can actually implant multiple ORI sites over here, and we can do some playing with the genome, but we have to actually reduce the complexity of the system, the CRISPR-Cas9 system. It is done in labs nowadays. In India it is actually done in huge amounts for different kind of purposes, but everywhere the basic of this CRISPR-Cas9 is same. It being, we have to take a genome, we have to know what kind of gene we want to silence, we have to make the mRNA of it, we have to incorporate into that, so the 20 nucleotide sequence I am talking about is going and binding to that, recognizing the PAM sequence. This Cas9 system is then cleaving it, the genome is silenced either, or otherwise we can add new gene using some target DNA. So this target DNA can be actually incorporated, okay. Homologous incorporations and all we can do, and that can actually help in making new kind of genomes from the parental genome. And this can be, if you are not planning to incorporate any DNA, then this double-stranded breaks can be repaired using the non-homologous enjoining, which can be random mutation type of, it can cause random mutation, and also is a error-prone kind of process. And also we have got homology-dependent direct repair, also known as HDR. So I'm concluding my video over there, because if you want to know more details about the CRISPR-Cas9 and how we actually implement it, I would actually ask you to like my channel, subscribe to my channel, and also share my content, okay. There are many people who need this kind of knowledge, and they are very talented young brains. And I, being non-talented, I try to teach something. I don't know how much I can reach, but I want you guys to just go and make them study these videos, and also ask them to study papers, research papers. Those are actually going to help you. And for knowing more about this CRISPR-Cas9 system, please subscribe to my channel and share my videos in YouTube, Facebook, Twitter, anywhere you can do. Just make it sure that our knowledge not only gathers up in our family, but also it spreads to different families, and they can learn as well, okay. So that's how I want to conclude my video now. And as I am saying that today we will having two different video lectures. One I am uploading, this is CRISPR-Cas9, and the other video I have not making it yet, but I will make it. That is will be the cell signaling, not cell signaling, sorry, that's a different project I am working on. I will be delivering the next video lecture, and it will be about the cell cycle checkpoints. Okay, and it's a very short video it will be, but please like, share and subscribe to my channel, and please help me increase my followers and also help me to learn more by making you guys learn. Okay, this channel is for you guys, and please, in case of any doubt, comment down below. You can directly mail me, mail address is given. Also, I have provided my website, so you can also pin your comment over there so that I can see it. And also, I would like you all to learn and grow. Thank you. GTFO. I'm not going to say the full thing of GTFO. Okay, you understand that because you are all guys are naughty. Okay, bye.